Alright, SBI4U, this is video number 5 for you. This is Thursday's video. We're doing the introduction to photosynthesis. Now we spent a really long time going over metabolism because it was brand new, this idea of like proton pumps and ATP synthase and membranes and things like that. But we're going to go through photosynthesis in maybe 3 days here. Because you already know a lot of the background of how life likes to do things, and life likes to use the same tricks. So quickly we're going to review basic human carbon processing, just a quick review of how it works for humans, and then we're going to jump into a brief overview of photosynthesis, of kind of the similarities and differences between the human version and the plant version of this stuff. Then we'll talk about plants and how plants actually support that with their cellular organization. They're very much organized. The leaves are very structured in order to support photosynthesis. And then a detailed look at the structure of the chloroplast, just like we had a detailed look at the structure of the mitochondria during our metabolism lesson. Yeah, that should be enough for today. So a lot of this is going to happen by comparison. So including the keep in mind up here, in both animals and plants, the electron transport chain and the carbon cycle happen in different places. Without further ado, let's get to our current topic, which is the carbon cycle of human versus the carbon cycle of plants. Jumping over to the document camera and taking away the formula sheet. Alright, let's do an overview of the carbon cycle versus the plant cycle. And my webcam is not active. That's interesting. I'm going to pause this and figure this out just a sec. Okay, I fixed it and my webcam's back, so let's get to it. Let's get talking about the mitochondria versus the chloroplast. So first off, the human carbon cycle, if you recall, there's the cell. This is the big cell, right? The cell. And then there's the mitochondria. And inside the mitochondria is mitochondria, and inside the mitochondria is that electron transport chain, which happens here, right? The electron transport chain, and then there's the ATP synthase. Right, ATP synthase, and the whole point was getting a bunch of H plus ions all gathered up in the intermembrane space. That intermembrane space, getting a bunch of H plus ions gathered up in there, and as they flow back through, those H plus ions release energy, and these pumps are the ones that get the gradient going again. So that's the electron transport chain. The thing that feeds into the electron transport chain, the thing that actually drops off the electrons, is NADH and FADH2. So just a really quick review of what it looks like. Right, NADH and FADH2, and that's making a bunch of ATP. That NADH2 will sometimes come in from the outside, and outside of this you've got glucose becoming pyruvate. Right, glucose becoming pyruvate, and pyruvate sneaking in to go through the Krebs cycle, and there's the Krebs cycle in there. But the Krebs cycle is separate from the electron transport chain. And the the ATP synthase is separate from the electron transport chain as well. So everything is separate, and we can kind of do something like this, that glycolysis is in the cytoplasm, that pyruvate oxidation and the Krebs cycle, right, these guys happen in the matrix. And that's where these two in the matrix of mitochondria, that's where the CO2 comes out. The CO2 comes out of this process in the pyruvate oxidation part. And all of that stuff, right? All that stuff, what it does is it generates NADH and FADH2, which leave that process and go to the electron transport chain, right? And that's going to make a bunch of ATP for us. That's the general carbon thing in plants. That the CO2, in animals, the CO2 in animals, in mitochondria, comes out in the matrix. It comes out in the matrix in this cycle. It's not linked to the electron transport chain at all. And as we're about to learn, the same thing happens in plants. So this is the human version, the mitochondria version, I should say, because plants do have mitochondria as well. What we're going to see, what we're going to compare it to, as a comparison here, we're going to compare it to plants. And in plants, it's not actually like there's the let's make a standard kind of big round, big square plant cell. There's the cell. And in plants, you've got the chloroplast. The chloroplast. And it has inside it, it has two membranes, just like the mitochondria. But inside there, what you're going to see when we talk about chloroplast is there's another membrane. There's another membrane. Compared to the human version of this, in the mitochondria version, the H plus builds up in the intermembrane space. Here, the H plus actually builds up inside this structure, which we call a thylakoid. It builds up in there. If it's building up in there, where do you think the ATP synthase is? And the ATP synthase is actually facing outwards. And so to make ATP, the hydrogen ions here, the H plus, actually flows out to make ATP. Right, that's the ATP synthase. It flows out into this this middle area. Right, it flows out, which means that the electron transport chain, right, the pumps of the electron transport chain that need to pump it in, are actually also kind of facing backwards. So one thing that you're going to notice a lot is that the photosynthesis thing has a lot of the same features, but just kind of backwards from the animal one. So there's the electron transport chain for plants. They have an electron transport chain. They use ATP synthase and a hydrogen gradient to generate ATP, but instead of being in the intermembrane space and coming back in, they're actually on the very, very inside space, and they're flooding out in the chloroplast. So that's one thing you're going to see. And just like in the matrix of the mitochondria, there's the Krebs cycle and CO2 comes out. What you're going to see here is in the matrix of the plant cell, there is a different kind of cycle named after a different person. There's a cycle here. It's called the Calvin cycle. And instead of CO2 coming out, CO2 comes in. So CO2 actually comes into this cycle, CO2. And the product of this cycle on the other side is G3P. If you guys remember what G3P was, it was one of the things in glycolysis. It was the middle thing. It was the middle thing in glycolysis, G3P, along the way. So this one, CO2 comes into the carbon cycle instead of coming out. It comes in, and the output is a carbon atom. Not a carbon atom, a carbon compound with 3 carbons on it called G3P, which can then go and become glucose. So that's the difference is, one, in the mitochondria, CO2 out, in the chloroplast, CO2 in. Both of them happen in what would kind of be the matrix, right? That's kind of the big inside space. So that's actually pretty similar, except this has a different name. So we're going to talk about those when we get to the names of these things. Just know this was just the general differences between the two. So in chloroplast, the hydrogen is going to build up on the very inside in this thing called the thylakoid and flow out, and then get recharged in the electron transport chain. Meanwhile, ours, we've just spent like 4 days covering it. So yeah, so there's similarities in that there is an electron transfer chain and there is ATP synthase and there is a carbon cycle. But on this carbon cycle, the carbon comes in and a complex molecule comes out. Here, the electron transfer chain is actually pushing stuff in instead of pushing stuff out. And the ATP synthase is driven by the gradient coming out of the thylakoid instead of coming back into the matrix like we have in our mitochondria. That's your basic differences. So looking at our learning goals, right? That was the first two. The brief review of human carbon processing and the brief overview of photosynthesis carbon processing. We're going to get way into that on Friday's video, talking about this cycle in molecular detail. I'm going to pause for a sec as I set up the next thing. Okay, I've moved over to the note now, and we're talking about now plant structures that support photosynthesis. And the keep in mind has changed to say that specific wavelengths of light can excite specific electrons on a molecule, and excited electrons have more energy and they can participate in an electron transport chain. Like in our metabolism, we need that high energy molecule, the NADH and the FADH2, to give the electrons those energies, but a plant can just get that energy from the sun. That's how they kick their electrons up their transfer chains, and as the electrons fall down, they're going to pump those H pluses. And then we're changing the current topic to plant structures that support photosynthesis. We're talking about molecular structures and physical structures as well. So quick overview of the reaction. You guys have seen this reaction probably many times by now that you're in grade 12. You see it in chemistry class, you see it in grade 10 science, grade 9 science. This is the overall. It's not balanced. It's typically set as 6 CO2 and 6 H2O makes 1 glucose and 6 oxygen. But the light is important for the energy. Because as we learned in metabolism, this is a higher energy molecule than this. So falling down the curve one way means you have to climb back up the curve the other way. You can't get around it. That's the first law of thermodynamics. Energy is conserved. If you're releasing energy going one way, like us burning our glucose, you have to put in energy going the other way. That energy comes from the sun. So structures that support this. You gotta think about this, the fact that the leaf needs, we've got like a bonus thing here, let's take that away. You can think about the fact that the leaf needs to work with this reaction here, right? It needs to be able to pull in CO2 and pull in H2O. It also needs to take in light. And then it has to store the sugar, and also it has to spit out oxygen gas. As we just talked about in yesterday's video, this is toxic, so the plant might not want to keep it around for too long. So all of this, everything in red, is there to support that specific goal. So first things first is plants don't want to lose water, right? Plants don't want to lose water, and that's the goal of this thing, which is the cuticle. The cuticle, it's a waxy covering, one of the rare examples of a lipid polymer. So yeah, a waxy coating here and here on the top and the bottom of the leaf. Some plants have thicker cuticles than others, and so they'll feel even waxier. Cactus comes to mind. Now, so that's the cuticle. It prevents water loss because it's like having a layer of fat that repels the water and keeps the water in. That water comes in through the xylem. The xylem is the water-carrying tissue in a plant, and it's all through the leaf, almost like a vein or an artery in your body. And the phloem takes away the sugar and other products that the leaf is making. So xylem brings in the water. And the last piece here is the stoma, which is a little opening in the bottom of the leaf, far on the other side from the sun, right? So it's not getting direct sunlight. And when it's open by these guard cells, air can come in and out. So oxygen can leave and CO2 can come in. Remember that one of the main goals here is pulling in CO2 and pushing out oxygen. So when these are open, the CO2 can come in and the oxygen come out, and the plant can breathe. We don't call it respiration like we would in humans, we call it transpiration. So let's put that word up there, transpiration. There's the word transpiration. Is a plant exchanging CO2, O2, and H2O with the air. Right. If you've ever been in like a place with a lot of plants, it gets very humid when it's daytime, because they are opening up these guard cells. These stoma are open, and they're pushing out water, and they're pushing out oxygen. So they need to open them to push out the oxygen and bring in new CO2, but the water leaves with it, and that's why it gets really humid in a place with a lot of plants when they're actively photosynthesizing. So that's the first piece. The xylem brings in the water. The stoma opens during the daytime when the plant wants CO2. The cuticle prevents water loss everywhere else. This whole thing is called a vein because it looks like a vein, like a human vein. Alright, now the actual light part, taking in the light. There's two types of cells that can take in the light. There's the palisade mesophyll, and the spongy mesophyll. I'm going to pause for a second, find a picture of a real palisade, so I can explain to you why those cells are called palisade cells. Just a sec. I found one. This is from Wikipedia. You can see just this fence thing. That's a palisade. That's called a palisade wall. So it's just a bunch of logs stuck up in a row. And now you can hopefully see why these are called palisade mesophyll cells, because whoever named them was reminded of those palisade walls. These are really good for the gathering light part because the light is coming down from the top. And as the light comes down from the top, right, the light is coming down from the top, it'll hit more of the cell because it's coming straight down. Right, the light's coming straight down, so it's a densely packed wall of stuff for it to hit. Right, if the cells were flat, the light might go right through them, but since they're like this, the light hits somewhere in the middle there. The light hits it somewhere. So those are the palisade mesophills. The spongy mesophyll also have chloroplasts in them, so they can also do photosynthesis, but they're spongy, and you can see they actually kind of look like a sponge. and their feature, the reason that they're spongy like that is to leave air pockets inside the leaf so that the CO2 and the O2 can be safely exchanged, and there's place to kind of store that gas that's building up and the water vapor that's building up. Because if this was densely packed with cells, you wouldn't have room for it, basically. So that's how the plant structures support the photosynthesis. And this is talking about the stuff that I was talking about earlier, where the CO2 is actually separate from the H2O. Just like in humans, the CO2 is separate from the H2O. CO2, Krebs cycle, and pyruvate oxidation in the matrix of the mitochondria. H2O created in the electron transport chain after the oxygen grabs the electrons. Let's talk about chlorophyll. You might have heard of chlorophyll. Chlorophyll is the main molecule that actually absorbs the light. It looks green. So anytime anything green in terms of a plant will typically have a lot of chlorophyll, and because most plants photosynthesize, they've got chlorophyll. Here's what it looks like. Here's what that's not chlorophyll. Sorry. Chlorophyll's main purpose is to gather that light energy, and we'll going back to keep in mind that wavelengths. You know what? I'm gonna pause while I do this. Okay, I'm back off pause. Keep in mind that certain wavelengths of light can excite electrons on specific molecules, and chlorophyll is that molecule. Chlorophyll gets hit by the right wavelength of light, the electron jumps up at a higher energy, and then they can start falling down and do electron transport chain stuff. And the electron transport chain stuff it does is going to make ATP, and it's going to generate this stuff called NADPH. So it's like NADH but with a phosphate on it. So here's the thing. Here's NADH. Here's NADPH. Here's the P. There's the phosphate. So it's a lot like the energy carrier, the electron carrier that we have in our bodies, except for in our bodies, the electron carrier drops things off at the electron transport chain. In the plant, the NADPH actually picks up electrons from the electron transport chain and brings them to the carbon cycle, to the Calvin cycle in the plant. Photosynthesis happens here in the chloroplast, so that's going to be our next topic, which is the structure of a chloroplast. I'm going to pause for a sec, and I'm going to, make all this the right thing, and I'll see you in a bit. Okay, I'm back, and we're on our last section, which is the structure of the chloroplast. So the chloroplast has 3 membranes and 3 different sections. This is a picture of it, but the arrows are kind of hard to show what we're looking at. So what I'm going to do is I'm going to jump over the document camera, where I've got the same thing diagrammed, but in a simpler way. And I'll pull up this one on screen just so we can see it, and we'll zoom in a little here to keep it on screen. So that's the thylakoid there. That's the chloroplast there, and this is my version of the chloroplast. So there's the rest of the cell going out that way, so the cytoplasm is on the outside. And please keep in mind it's got 3 membranes and 3 distinct sections within those membranes. So out here is the cytoplasm. In here, it's a space between 2 membranes, so it's an intermembrane space. But unlike the intermembrane space for mitochondria, nothing important really happens there. It just has two membranes for some reason. So these membranes have names. This one, just like in the mitochondria, this one is called the outer membrane. Outer membrane. And this one is called the inner membrane. Right? And between them is obviously the intermembrane space. Then you've got this middle section here, kind of, this stuff in here. All of that has a name. In the mitochondria, that's called the matrix. Here, it's called the stroma. The stroma, that's the space in the chloroplast. So that's the stroma. It's basically like the matrix of the mitochondria, but the chloroplast has this up its sleeve. It's the another space inside. So this is the stroma. In here, there's another space with different stuff in it, and that space is called the lumen. That's the lumen. So that's what you're seeing over there on the picture, on this picture here. You see the stroma that's trying to point to that space. The stroma is trying to point to this space. They don't actually have a name for the inside on this diagram, but now you know it. It's the lumen space. Sometimes it's also called the thylakoid space, because this thing, one of these guys is called a thylakoid. Thylakoid. They're flat discs, and they kind of look like a coin. A lot of people compare them to coins. So that's a thylakoid. And a stack of thylakoids, so like more advanced plants will have a stack of thylakoids, one on top of the other, to help them photosynthesize. A stack of these is called a granum, which is a stack of thylakoids. Stack of thylakoids. So a lot of terminology here compared to a mitochondria. There's more to talk about because there are three distinct spaces: the intermembrane space, the stroma, and the lumen. And there are three membranes: the outer membrane, the inner membrane, so the outer membrane, the inner membrane, and then this thing, which is the membrane that surrounds the thylakoid, which is called the thylakoid membrane, because biologists aren't clever. Or maybe they are clever because it's easy to remember. Maybe that makes them more clever. Who knows? So yeah, that's the thylakoid membrane, is the thing that surrounds the lumen, that makes the thylakoid, is the thylakoid membrane. And that's the basic organization of a chloroplast, and that's what you're trying to show you on that diagram. But that's what a real chloroplast kind of looks like. This is the kind of simpler version. I'll stick a picture of this on the SBI page so that you guys can see it. And briefly, just to review, in the chloroplast, the H plus is going to build up, right? The H plus builds up inside the thylakoid. H plus is going to build up inside that thylakoid. And it wants to come out. It wants to come out, right? It wants to come out. And the only way that the thylakoid lets it out is through our good friend ATP synthase. ATP synthase. right? So the ATP synthase sits on the thylakoid membrane now, and it is going to be the turbine that those have to go through, that the H pluses in here have to go through to come out. And we're also going to see tomorrow, tomorrow's lesson, Friday's lesson, that there's that cycle in here, that carbon cycle, which happens in the stroma. There's a carbon cycle here in the stroma, and CO2 is going to join that cycle, and G3P, which is a precursor to glucose, is going to come out. And that G3P will eventually become glucose. Right, 2 G3Ps will become glucose, or any other sugar that the plant needs. Now that requires energy in the form of ATP. So that takes ATP minus ATP to make this run, because you're building up the G3P. And it also takes this NADPH stuff, which is a lot like NADH, but it's NADPH. That's generated up here. The NADPH is going to get generated at the thylakoid, and then it's going to kind of come in here and join there. So they are separate. They're separate, but they're linked, just like the CO2 part of our mitochondria, the Krebs cycle, is separate from the electron transport chain. They happen in different places, but they are linked by molecules, by the molecules ATP and by the molecule NADPH. So that's your intro to photosynthesis, and that's what we're going to talk about in detail tomorrow in Friday's video. Yeah, thanks for paying attention, and I'll see you in class. I'm sure you'll have lots of questions about this stuff. Bye.